Hey guys, welcome. Do you know what's worse than finding a bug in your spaghetti? Having some viruses in your pills. Well, first let's meet our team. Here's Jason, here's Arzoo, and I'm York. Together, we're making a computational pipeline to detect viruses in Chinese hamster ovary cells, or CHO cells. So why CHO cells? Because CHO cells are widely used by biopharmaceutical industries for protein therapeutics manufacturing. And protein therapeutics, you can just imagine that as some kind of drug you're gonna eat at some point in your life. Well, I bet you already noticed how bad it would be to have some drugs with virus in it. And then you should probably start to realize how important our project could be. Well, now I'm passing on to Arzoo.
Hello, I'm Arzoo. Being mammals, Chinese hamsters have the potential to easily make proteins that are as complex as ours. But this also means that they can carry viruses with the potential to infect us. Protein therapeutics manufacturers try to keep their cell lines free of infection so that viral particles have less of a chance of slipping through into the final product. In order to help detect viruses in CHO cells, we've made CHO Detect, which is an efficient and easy to use bioinformatics pipeline. Of course, you may have guessed that there are other pipelines out there, but these pipelines have their limitations. Some require more than a terabyte of space, others take extensive knowledge to use. What sets our pipeline apart is that it's designed for CHO cells. Now Jason will talk about the potential impact of this project.
Thanks, Arzoo. The ability to detect viruses with just a simple RNA sequencing sample has the potential to change how pharmaceutical companies conduct their day-to-day operations. Normally, if a lab's production cell becomes naturally infected with a virus, then the lab may need to sterilize all of their equipment to quarantine the spread, which often means halted production for weeks or even months in the most severe circumstances. This causes a serious shortage that, if other sources of the drug can't keep up with the demand, could result in massive losses. The most probable way a viral infection occurs is through the introduction of a new sample. Whether for testing purposes or to replenish stock, new cells brought to the lab always have a chance of having a viral infection. This is where our software pipeline CHO Detect comes into play. Simply extract the RNA from the samples and sequence it to get the files needed for our program. After that, you just have to run it while you're doing something else, and once it finishes, you'll have a set of results that tell you if your cells have a viral infection, and if so, what viruses were found.
We would like to thank Dr. Lewis and all the collaborators who helped us along the way of our project.